Thank you very much. Yes, well, it's a big pleasure for me to be here, and thank you very much for the invitation and everybody for the interesting presentations and also the delicious coffee. I'm a physicist by profession, and actually my contact with coffee, first contact was in school, where a physics teacher would brew coffee during class for himself because he was addicted and all the students. And this, I think it didn't increase the, the creativity of the discussions, and which made his lessons so interesting. So yeah, after that it helped me through university and my years as a service engineer. So more than glad to be here. So yes, I'm working at the Kurt Engelhorn Centrum of Archaeometry, the CESA, and you might ask, what is that? And archaeometry is about investigating the chemical and physical properties of a sample provided by archaeometry, so archaeology. So for example, if, I don't know, people dig out a skeleton or whatever, and they can send it to us, and we can do several things. For example, we can do the famous radiocarbon analysis to determine the age of the person or the animal who has died there. That's with the big accelerators. You can visit and see if you like. Or we can do isotopic studies to determine what the diet of the animal or the person was like. More meat, more fish, more vegetables, which kind of plants, C3, C4, stuff like that. And also we can do all kinds of metal analysis to determine okay, where might the gold from the ornaments of the Himmelsscheibe von Nebra come from, and stuff like that. So, right, and since we have the instrumentation and the numbers of archaeological samples is somehow naturally limited, you can't dig out stuff forever in quantities. We are always excited and interested to get new applications for our instruments. And Herr Schwarz was so kind to provide us with a big box full of coffee and a fantastic data set from different countries, different varieties, different fermentations, everything. So, and we're playing around a little bit with that to see if we can do some authenticity stuff maybe with it. So as you might know, isotopic analysis is a big thing in food authenticity. You can determine where does the wine come from, is the honey adulterated with sugar, is the vanillin made from vanilla or from pure synthetic chemistry and stuff like that. So there's a huge framework for this technique, and there have been studies on coffee, but yeah, apparently there's no agreed on framework how to judge. So, and yeah, what we think we might do is, together with Herr Schwarz and his fantastic samples, to build up a small database, understand what we see, and or maybe contribute to an existing database. So let's see. So the first thing we can do with a sample of coffee beans is elemental analysis. And elemental analysis is just a very simple thing people have been doing for over 100 years. It's just basically you burn the sample, and then all the carbon in the sample transforms to CO2, which you can analyze. All the nitrogen in the sample is nitrogen, which is a gas. All the oxygen, all the hydrogen, everything, so it falls apart, and you can, as a first step, you can detect the elemental composition of a sample. And the main elements are, of course, carbon, hydrogen, nitrogen, and oxygen in everything that grows and lives. And so, for example, this is the molecule we are here. That's the reason, because this is caffeine, as you probably all know. And they're all there. And we did this as a first tryout with a sample of coffee beans, and what we got is this. And don't be too strict on the numbers. It's, there's some uncertainties, but it's mainly, coffee is mainly just that. Yeah, nitrogen, hydrogen, oxygen, and carbon, and some minor constituents like sulfur and strontium and whatever you find everywhere. So, okay. In the literature, you can find people already making some results out of only this elemental composition. So, for example, the difference between Arabica beans and other beans should show up in the elemental composition already, if I remember correctly. And so this is just a first start. We regard it sometimes like as a little bit as a waste product. And the next thing then will be more interesting, and we split up all the nitrogen and the carbon and the oxygen and the hydrogen we find in the coffee beans into its isotopes. And I just want to repeat quickly what isotopes are in case not everybody is familiar. So you know that an atom is built from a nucleus and the electrons, and in the nucleus you have the number of protons, which defines which element you're looking at. Is it carbon, is it nitrogen, or whatever? And also you have the neutrons in the nucleus, and these define the mass of the nucleus together with the protons. And this then is called an isotope. So you have, for example, in the case of carbon, you have 6 protons, and normally you have 6 neutrons, which add up to a number of 12 in the nucleus, which is carbon 12 then. And but you have also stable isotopes with a different number of neutrons. So in the case of carbon, you also have carbon 13, which is carbon and behaves like carbon, but is a little bit heavier. And with a mass spectrometer, just a really old-fashioned, simple sector field mass spectrometer, you can split up the carbon isotopes, carbon 12, carbon 13, and then for the coffee beans, you get a number like this. So you have about 1% and then a lot of digits after the comma of carbon 13 in the total carbon of the coffee bean. And you can do this for nitrogen, for hydrogen, for oxygen with the same instrumentation, relatively easy, and you get all these numbers. And these numbers are very small, as you can see, and also they have a lot of digits. That's for a good reason, because the variation is only in the last digits. So that's the reason why isotope ratios are often expressed not like it's done here, like an absolute percentage, but in a relative deviation from an international standard. So carbon is presented into relative deviation to stone, called the Vienna PD-Belemnit standard. And so this coffee bean would be 27.8 per mil lighter in carbon-13 than that stone standard. So you can be, negative means you are below the standard, positive means you are above the standard, and we call negative values lighter and above values, positive values, heavier, because you have more of the heavy isotopes.
That is just to interfere, like we said yesterday in medicine. Sometimes things positive are negative and negative is positive. So when you gained weight, you can say, oh, I look a little bit positive today.
I feel positive, thank you. So these are just the results. You don't have to make anything of that now. This is just the results we got for a coffee bean. Carbon, nitrogen, hydrogen, and oxygen. And now, of course, is the question, what do these numbers tell us, or what can they tell us if we build up a database? And with our playing around, we rely on the literature, a PhD thesis from the University of Lisbon, from Christina Hansen's group, a lady named Carla Rodriguez, and she put together a lot of isotope measurements in coffee beans. The coffee beans were provided by the company of Nova Delta, and they did all kinds of things with them, and they have the same instrumentation that we have. So that's why we stick to this literature and this try to follow the recipe. And for example, one very interesting thing which you can do is geographic origin. So everything in nature, every chemical or physical process has a certain slight effect on the isotopes involved. So for example, if water evaporates, you will see a certain shift in the hydrogen and oxygen isotopes compared to the ocean, where it evaporates. And then when the cloud of water vapor travels over the land, it will form raindrops, and this again has an isotope effect. So the rain has a different isotope ratio than the cloud. And then the rain falls down, and the cloud changed now moves on. And that's why you have, for example, for hydrogen and oxygen, you have isotope patterns like isotope landscape maps all over the world. And you can, for wine, this is done by looking at these two isotopes. You can tell, okay, this sample looks like it came from this or that geographic origin or not. And also very interesting if you take sulfur isotopes into consideration. Sulfur is in the ground, but it ends up in the coffee bean. They contain sulfur. They did a very interesting study here where they used a set of isotope tracers, including sulfur, to track down coffee-producing regions in Hawaii. So in a very small environment, they were able to track down certain regions because the sulfur somehow correlated with the distance to the volcano and stuff like that. And also for geographic origin, so you have oxygen and hydrogen for water, but very useful for the land is strontium. We can't do this with the machines I'm talking about now, but we also have strontium analyzers in a different lab. So that's something we could potentially do as well. So geographical origin is a thing that you probably can do quite well with isotopes, but also other things like carbon and nitrogen isotopes are influenced by the way the plant grows. So if it experiences high temperature or dry stress or whatever, the carbon and nitrogen isotope ratios will change. And they're probably different for different types of coffee plants, so Arabica and other stuff, or even varieties of Arabica. And so we have a whole playground, at least 4-dimensional parameters room. And we just tried it out. And so that's the first thing was we tried we threw in some coffee beans from Brazil, from the Dutra farms, and we threw in some beans from India, from the Badra estates. And you can see a lot of things are very similar. So nitrogen content, carbon content, hydrogen, oxygen content, all very similar. The below values are the standard deviation, by the way. But you can see that, for example, nitrogen isotopes are different, 2.9 versus 2.1. Or carbon isotope ratio is different, -27, -29. Hydrogen and oxygen also show a big difference. With hydrogen you have to be a bit careful because the element is tricky to measure. But -71, -68, and for oxygen, a really big difference, which is expected because of the rain pattern stuff I talked about, 23 to 20.8. So this is just, of course, this is not a result, but just an outlook. But this seems to be working quite well as expected. So our results are all in accordance with the published values of Rodriguez, for example. And then the next thing we tried to look at was different varieties of Arabica. This is again Brazil samples, and everything else was kept constant. The same fermentation. By the way, this is green beans. We started with green beans to be as close as possible to the geographic origin and the growing conditions, of course. And with the different varieties of Arabica, again, many things look very similar. But for example, one thing that might be interesting, the oxygen content of the beans looks a bit different for these 3 varieties. I know nothing about, but you probably do, Herr Schwarz. And also the nitrogen content was a bit different for the Catigua, whatever that means. We have a difference in nitrogen isotopes as well between the 3 varieties of Arabica, and but the rest looks more or less the same. But this is it points in the direction that you might be able to differentiate between different coffee varieties with carbon and nitrogen isotopes. I should mention that since this is our first attempts and just playing around, the standard deviation of the results is quite high because we found out when we looked at the results, unfortunately, that apparently, what we do is we take green coffee beans, put them in a mill, and then take some of the powder we get and put it in a tin boat, fold it, and throw it in the machine. That's a really small ratio, that sample to the whole set of coffee beans. And apparently our sample was just not big enough, and because the mill was not working fine enough. So next time we will use bigger sample sizes, and then all these standard deviations will come down a lot. So these are much above what the instrument can do, and we should reach better results and then better differentiation when we do this next time. So so we have differences in varieties, we have differences in the country of origin, In the data set of Herr Schwarz, we have lots of different fermentation techniques of lots of different samples. And we thought, okay, now if we have the green bean from a certain country, it looks like we could make something out of the isotope values to track down the country and stuff. We need to check what happens afterwards with the bean, and does this change the isotope ratios? Because then maybe, it could be that you have these 3 varieties and then you ferment them in a different way and then they all look the same, or they change around, or whatever they do. So we needed to check fermentation. And so this is the same coffee, different fermentation, and to our pleasant surprise, a lot of things looked very similar. So I think there's no significant difference due to fermentation in the elemental content or the isotope ratio, which is very interesting. So this wouldn't harm our results and our tracking. And finally, what we thought would be very important, that's what, there was a question about that for the last talk, the gap between green coffee beans and roasted coffee beans, because of course you have the green coffee beans, they have the signal from the origin and from the plant and everything, but what you buy in the store is a roasted coffee bean. And to us, the question was very important, is there a link, or with different roasting can you do all kinds of things to the isotope ratios? And so we tested that. So again, in the data set, we have roasted beans and green beans from the same, so basically, the same coffee, green and roasted. And what we saw, well, of course, some things have to change. So because of the coffee is dried during roasting, of course, hydrogen and oxygen content will change. But they don't change so much because we need to dry the green beans before we analyze them. So don't be surprised. Herr Menzel told me that the coffee beans lose something like 10 or 20% of their weight in water. So you don't see this here because we dry them. But still you see some change, and also you see some change in the other ratios, because everything changes when things like oils and water and everything just evaporates. You have a different structure afterwards. So that was not surprising at all. The very interesting result is that the nitrogen isotope ratio is identical in the green bean and the roasted bean, which is very nice. So we have one thing that is absolutely robust. The carbon signal, you can't really tell. I mean, it's close and the standard deviation is high, so I would expect that it changes at least a little bit, because you lose carbon because of the oil, which is evaporating. But on the other hand, all the things that happen, the Maillard reaction and everything that happens inside the bean, when you roast it, happens inside the bean, so it stays there. So if you look at sugars or oils or proteins or whatever, they all change around during the roasting. But if you look at the total bean bulk, then probably you don't see much change, because it just goes around from one molecule to the other. And we expected all kinds of changes, but this actually looks very promising, that you can still make sense of the carbon isotopes after the roasting. Hydrogen and oxygen, again, show the biggest difference, but that's to be expected. And so this was an average, and we looked a little bit further into the things. So now we just compared the differences. So we have one sort of coffee with that fermentation, just for completeness. And now what you see here is the difference between the roasted beans minus the green beans in the signal. And the dark colored green and red is a very, very robust signal, so difference is bigger than 2 times the standard deviation. The light green and orange is a bit questionable signal between 1 standard deviation and 2 standard deviations, and in yellow we have things that are just look like noise with our precision that we have now. And okay, what we can see is nitrogen goes up a little bit in content, but not much. Carbon goes up in content significantly. Hydrogen doesn't change at all, but that's maybe due to the instrumental resolution at the moment. And oxygen goes down. Oxygen content is lower in roasted beans, a robust signal. And for the isotopes, the really interesting thing, here again, you can see nitrogen is more or less unchanged. You have not one robust signal for nitrogen isotopes. Oxygen, on the other hand, goes up. So the roasted coffee beans are heavier in oxygen isotopes than the green beans, which is in accordance with the theory and the expectance, because normally if something leaves a certain entity, like a coffee bean, then normally the light isotopes leave first. So the remaining isotopes, the ratio changes to more heavy if the light isotopes leave. So this is perfectly what we would expect. It still tells us that we might be a bit careful when we do interpretation of isotope ratios of roasted coffee beans, because the oxygen might be changed. For hydrogen, again, well, it looks like it goes down, but it's a bit questionable, as I said, due to the instrumental precision at the moment. And for carbon, it goes mainly up, and the interesting thing is, for one sample, it goes down, which is not expected. As I said, it should get heavier. Now the question is, if this is just an outlier or maybe a sample mix-up, or is this maybe something which is characteristic to the roasting technique or the coffee beans or whatever. But yeah, this is only just the beginning, just a lookout, an appetizer, and we're excited to continue this work. And finally, no coffee presentation without a coffee picture. And with that, thank you very much for your attention.
So the picture, thank you so much. The picture was a reminder of a standard coffee in Germany.
Yeah.
No, I'll never comment on coffee pictures, I learned. Okay, so I didn't do this. Okay, so
this picture was actually coffee out of a vending machine.
Yeah, okay. Good news that Mr. Kashevi from the Vending Association has left the building. So, the questions. Here. No, wait, the microphone is here. Sorry. Yeah.
Hi, a very nice presentation.
Oh, thank you.
I'm very interested because during the Maillard reaction, as you said, during the roasting occur a lot of things, but during the fermentation also.
Yeah.
And then do you have like a way, like the nitrogen didn't change with the oxygen and so on, with this figure number that you have for the other elements that you have in the structure of the compounds, you already has like a pattern, typical one, just to say here you have more caramelization of more Maillard reaction, you know, to identify the pathway of the modification that you have, or you can just to make an identification between the light one, the dark one, or the Arabica one from the Robusta one. I mean, I don't know what is the application that you can give in the stage you have your investigation just to make an identification between the different roasted materials. I was clear or no? Or it was too long, the question?
If I can jump in and maybe simplify,
Yes.
let me try to rephrase.
Yes.
We learned from Professor Kohler to rephrase the question.
Yeah.
So I think, please correct me, the question was if in the moment, at the stage of your research, you can identify between species, varieties, or also between intensities of
Processing
a roast.
condition, yes. If you can just to say, this is
Italian
fermented.
roast, German roast, or
that's
even
right.
Borneo roast.
Perfect, yes. This is the question.
Well, well, the simple answer is at the moment we can't differentiate. I mean, we can't differentiate like for sure between anything, because this is just, we analyzed, I think, something like 50 samples now. This is just the beginning. It's just a starting point, and we were just playing around with the method, see if we can reproduce the results from the literature, and get a feeling for the method and see if it works. And it seems to work fine. Yeah, absolutely. I think. I mean, we saw differences even in Arabica varieties. Small, but they should be significant. But we have to check if these change, if, for example, that Arabica coffee comes from another country, or, you know, so.
My suggestion is just to try, if the other presenter allow me, is just to make a comparison between volatomics and this method that you are proposing, because see that the volatomics allow us, as far as we know, because a recent publication that we have with the Slovakian research, that even the green beans can be differentiated, look into the volatomic results. And then probably just to make a validation of your method when you have probably Professor Schwab can give you more samples just to do more analysis, and probably it's nice just to compare the data from volatomic to yours in turn, because sometimes volatomic is very complex, and if yours is a simpler method, probably for the company is very good just to establish a standard method, but for validation I think that you need a stronger analytical stuff, and probably this is an advanced analytical stuff that you can use it just to make the comparison. This is my suggestion. I don't know, and it may be a new collaboration if you allow me just to say that.
And yeah, that's actually what we do. So we have been sending around the samples from same things to at least 12 different universities and centers, and we all ask them to do their analytics on it, and then we bring back all the data together. So we believe we are not more stupid than we were before. And we believe that connecting those data is in the beginning confusing. You remember there, yeah. We also learned about what is confusion and what is a complex problem there. So you see that everything comes together, and maybe Dr. Lachenmeier gives us a new insight.
Yeah, I absolutely agree with Dolores, and it's not called confusion but fusion, data fusion. I think Jan remembers well, we did this with wine analysis. We actually combined the isotopic values with our NMR analysis in basically one multivariate analysis, and you get a single model from this data fusion. I believe it's also Philip Weller's specialty to do something like this, and then you get much more robust separation of the groups than if you would look at each single method. And especially the isotopes and NMR, they are complementary, so you do not measure the same but so different information that is very, very well separates. If this is economically viable, and currently it's on an experimental stage and not validated, it's not accredited, this analysis, and it gives you all kinds of problems if you need a accredited, validated result for going before court. If you really find a deviation that your Hawaiian, whatever coffee, strange, blue mountain thingy is not the thing, we believe it in our analysis, but we still probably can't go currently before court because, as you said, it's still in experimental stages, those kinds of analysis.
So then we would just have to prove that it was deforested and then it's illegal.
I might add, if you allow me, that this is just so the differentiation between the coffee varieties, it's just out of interest. I think that we do that. I think with NMR, this is a well-established technique, and it's also found in that PhD thesis. They also did that. I think it's just more practical to do it that way.
Hi. Hello. How many samples are you doing for each variety, each cultivar?
So we have a data set of about 200, 300.
For each cultivar, okay.
Well, let's call it data points. So little bags of coffee, and with their metadata, and each bag is something like 40, maybe coffee beans, and yeah, we put those in a mill, and then we measure in triplicates.
Okay.
That's what brings the standard deviation.
Yeah, because I was interested in your standard deviation.
It's way too big. The instrument is much better.
We just analyze traditionally chlorogenic gases in the same plantation for 5 years. If we saw like different cultivars, you would think in only 1 year, you would think, oh, some of them were really great, full of chlorogenic acid. But if you analyze all of them for 5 years, you would see that in the end, they were all the same, you know, just doing the statistics. So if you, and you have also the botanical garden, you can even do the same same samples for different crops, different years. So you would have great data there.
So I see my trap is working on you guys. You start to think in cooperative models.
Yeah, I'm sure there will be big. There will be big signal changes from year to year, because the rain pattern changes, heat, everything. So we haven't done that yet, but I'm very, very curious about that. Yeah.
Thanks very much for the great presentation. It's really, really impressive to see. And I'm completely in love with isotopes still. I did that in my former life as well. What I'm still surprised of is that if you consider what happens during biochemistry, the whole associated processes, decarboxylation, evaporation of water, formation of water from chemical processes, not only contained water, that still those differences in the isotopes, in particular for carbon and oxygen, and also nitrogen, don't differ that much. Do you have an explanation for that, or an idea what that happens?
I remember we talked this over at Café Consulate when we had this meeting, and we hadn't checked it, but everybody was sure that you would see huge roasting signals. And it came as quite a surprise, and also in the Rodriguez papers, they only analyze green beans, probably because of that reason. No, it's a total surprise. My only explanation is that well, the coffee bean itself is quite closed. And all the chemistry that happens inside stays inside. And if you analyze the whole bean, it averages out. It's still.
Would you think it would be interesting to maybe do a separation of the lipophilic and the water extractable fraction and take a look at that, because that might be a summary effect what you're observing, right? Because a couple of substances may be building up, a couple of substances may be degrading. So on average stays the same, but that's not necessarily chemically reasonable, right?
Absolutely, that's the charming thing about this. Let's call it kind of not primitive, but simple analysis. You don't care about the compound-specific stuff, what's going on there. You just take the whole sample, put it in the machine, and just do a bulk analysis. This is many things you don't learn anything about with this approach, but it's surprisingly useful still for other things, because you can see you don't get lost between the trees, or what's that saying with the wood and the trees and what's that in English?
You can't see the trees because of the forest.
Oh yeah, yeah. And this is just, looking at a forest from above.
So I think this is a great word to lead us over to the coffee break. And so my last comment on that, I always shoot from left to right and up to down. This might be a good point also in the future, the isotopes, to pull in meteorologists. I know we have a victim here in the room, a potential victim. He doesn't know that I already kind of try to swallow him for future things, because meteorologists with your models might also find where first the heavy rain goes down with more deuterium inside and then help to whatever and climate.
They have models for that, yeah.
Yes. Have a nice coffee. Thank you so much.
Thank you very much. Oh, yeah, and And so if anybody's interested, I'm happy to show you around in our lab basement after lunch or whatever time is convenient for you.
Yeah, and I will sit down there with a cat
to
on
me.
my lap, and you'll be reminding of a James Bond video. I'll speak with a very strange accent then to you and say, "Welcome, Mr. Bond." Okay.